Bueno, buenos días a todos, a este seminario del curso de Frontiers in Genomics. Hoy tenemos la fortuna de tener a la doctora Anjana Rao. La doctora Rao obtuvo su doctorado en biofísica en la Universidad de Harvard y después fue profesora en Harvard Medical School hasta el 2011. A partir de ahí se movió a la Jolla Institute de la Universidad de California en San Diego, y su investigación ha estado enfocada en los últimos años sobre la regulación de la expresión genética usando células del sistema inmune, células troncales hematopoyéticas y células troncales embrionarias como modelo. Su laboratorio purificó y caracterizó a nivel molecular las proteínas TET, las cuales son desmetilasas del DNA. También en estudios recientes, su grupo ha definido el papel de los factores transcripcionales NAFT en relación a su actividad antitumoral y ha explorado los papeles de las proteínas TET y la oxidación de metilcitocinas en el sistema inmune y también en el sistema hematopoyético y en el desarrollo embrionario. La doctora Rao tiene numerosos reconocimientos y premios. Ella es miembro de la US National Academy of Science, the American Academy of Arts and Sciences, the American Association for the Advancement of Science, y recientemente obtuvo los Finding Investigator Award from the National Cancer Institute de la NIH. A su vez, ella ha formado más de 100 estudiantes y postdocs, los cuales muchos de ellos son investigadores independientes muy exitosos. Y recientemente es cofundadora de una compañía muy interesante que se llama Calcimédica, en el cual están estudiando inhibidores de canales CRAC para tratar enfermedades inflamatorias, incluso en la actualidad el COVID-19 también. ¿Sí? Entonces, thank you very much, Anja, to be with us. It's a pleasure to have you with us again. I think this is the third time that you are participating in the Frontiers in Genomics course. And well, now you can share your window at any time, and I'm sure we are going to enjoy your talk.
Okay, thank you very much for that introduction. I'm going to turn off my video briefly while I give the talk and then turn it on again later. So this time, today I'm giving a talk that is not about TET proteins, but rather about the cancer immunotherapy angle that we are doing, trying to figure out. Okay. Perfect. Yesterday I talked about this wheel that Hanahan and Weinberg have shown, which is very, very popular. And we're working on two things: TET proteins and genome instability. And the second thing is to avoid immune destruction. And everything I'm going to be telling you is a collaboration with Dr. Patrick Hogan, who is a long-term collaborator. So the story began a long time ago when Patrick and I cloned the NF-AT transcription factor. And we already knew, actually, that NF-AT went into the nucleus. It's a calcium-regulated transcription factor. So we already knew that it went into the nucleus when T cells were treated with calcium ionophores. We also knew that when cells were treated with PMA plus ionomycin, they actually got together, NF-AT got together with a different unrelated transcription factor called phosphin-Jun to form this upper complex, which is the NF-AT phosphin-Jun complex on DNA. And we then collaborated with Steve Harrison and Lin Chen to crystallize this NF-AT phosphin-Jun complex on DNA. And the question immediately became, since we knew that NF-AT could bind DNA either by itself or together with phosphin-Jun, does NF-AT have different biological functions with and without phosphin-Jun, which are together called AP1? So this is a very simple-minded schematic of the T-cell receptor signaling pathway. The T-cell receptor connects to PLC gamma and gives you both IP3 and diacylglycerol. The IP3 part of the pathway allows calcium to enter the cell through crack channels and sends NF-AT via calcineurin activation into the nucleus. Through the co-stimulatory receptors, they don't do anything to calcium, but they do amplify the diacylglycerol and PI3 kinase pathways. And in doing so, they turn on these transcription factors, phosphin, as well as NF-kappa B transcription factors that turn on target genes involved in effector function, cytokines, chemokines, and so on. So the way we are thinking about how NFAT with and without AP1 works is that we already know that NFAT in primary CD4 T cells, as well as in primary CD8 T cells, goes into the nucleus in 15 minutes and then stays in the nucleus for long times. In contrast, if you look at the AP1 components, and there are 7 AP1 family members, phosphin family members, they come up, this is a northern blot, and then disappear. So they're only activated very transiently. If one now looks at, puts these two curves together, you have NFAT without going to this for a long time, AP1 being transiently activated, and therefore the NFAT with AP1 program, which contains about 1,000 T cell activation related genes, is turned on briefly and then shut off. And the reason it's shut off is because phosphin and NF-kappa B and so on are potent tumor oncogenes, as you know. However, when we looked by microarray at the NFAT without AP1 program, we found that the NFAT without AP1 program only contained about 150 genes, and these were exhaustion related. I'll tell you what exhaustion is in a minute. Basically, it's a negative feedback program where the T cell turns on a lot of inhibitory receptors. And these are just the names of some of the inhibitory receptors that will come up again and again during the talk. So PD1, CTLA4, TIM3, and LAG3 connect to the inside of the T cell to turn on different kinds of negative signaling pathways that include tyrosine phosphatases, diacylglycerol kinase, which will remove the second messenger diacylglycerol, E3 ubiquitin ligases, which actually chew up things, signaling proteins that are close to the T cell receptor, and as well as transcriptional repressors. And so this was work done by Fernando Marcion, who is now a professor at Albert Einstein. And this is what I've shown you about the T cell activation program. What we decided then was that the T cell exhaustion program, called Anagir exhaustion, actually was turned on by NFAT without AP1 and included all these negative regulatory proteins that I just talked to you about. So to try to address the question of whether we could make a reagent that would turn on the Anagir program without turning on the activation program, we generated this mutant protein called CRIT-NFAT1. And this was done by Fernando also, with the help by looking at the structure from Lin Chen and Patrick Hogan. And what we then did was to mutate 3 residues in the NFAT phosphin interface that do not affect the binding of NFAT to DNA. And these residues are arginine, isoleucine, and threonine, which gives the mutant the name RIT. And we also made a constitutively active version of this that went into the nucleus, even without calcium entry, so that it was constantly in the nucleus. And the question we asked was, does that turn on the inhibitory program? And indeed it did. This is a dye dilution assay where we're looking at CFSE, which gets diluted by half with each cell division. And so when control T cells are CFSE labeled, these are cells that have been transduced with a control retrovirus, you can see that then in the second activation, they proliferate again, and they make plenty of interleukin-2. In contrast, the NFAT-RI cells transduced with this mutant protein really don't want to proliferate. Very, very small numbers of cells go into the cell cycle, and IL-2 production is almost completely shut off. So we decided then that in this paper, that NFAT without AP1 promoted the attenuation of T cell receptor responses. And that result sort of sat around for a while we decided, is there a biological program that we could use this protein in? And so at the time, about 5 years ago, everybody was getting excited about what is called T cell exhaustion. So in antiviral responses, and this is work done by Rafi Ahmed and John Werry and people like that, naive T cells are exposed to antigen, make a lot of effector cells. The effector cells clear the antigen, and we're left with a pool of memory T cells that are able to respond to a second antigen exposure with the same antigen. In contrast, however, if the viral infection is chronic, or such that the antigen is very persistent or the co-stimulation is poor, the NFAT-AP1, NFAT alone pathways are turned on, and the cells go into what is called an exhausted state. And the hallmarks of these exhausted or hyperresponsive T cells are, again, that they express these inhibitory receptors. They show decreased cytokine production, and they use key transcription factors in a different way. And this turned out to be very important for cancer immunotherapy in general, because, as you know, exhausted cells can be treated with antibodies in the body in patients, with antibodies to some of these inhibitory receptors, particularly PD-1 and CTLA-4, and those exhausted cells are turned back into effector cells. But what I'm going to be mostly talking to you about is a different kind of therapy where CAR T cells, chimeric antigen receptor expressing T cells, or activated cells of a different kind are put back into the body such that they boost the pool of effector cells, and you get antigen clearance, or tumor clearance in this case, a little more efficiently than usual. But even those cells that are put back into the body end up going through this exhaustion program, the hyperresponsive program, because that's a basic program, basic negative feedback program in T cells. So we asked, does our reagent NFAT without AP1 contribute to T cell infection? And this is a paper that was published more than 5 years ago, so I'll just summarize it. Basically, what Gustavo and Renata from Argentina and from Brazil, respectively, retrovirally introduced a CRIT into CD4 and CD8 T cells. And this was intended to initiate the inhibitory program before the activation program could even get started. And what indeed we saw was in vitro, we saw decreased cytokine production, just as Fernando had seen previously in CD4 T cells. We saw increased expression of inhibitory receptors, decreased tumor rejection in a mouse model in vivo, and decreased ability to combat Listeria infection in vivo. And it turned out that they resembled exhausted T cells by transcriptional profiling and ATAC-seq. And characteristic of the exhaustion program is upregulation of these two families of transcription factors. First of all, the inhibitory receptors, this encodes TIM3, this encodes PD-1, this encodes LAG3, are obviously turned on in the cells expressing the CRIT, NFAT1, compared to the DNA binding mutant. But these transcription factors are turned on as well. NR4A was a family of orphan nuclear receptors, which some of you may know as Neur77, Neur1, and Nor1, respectively. And TOX and TOX2 are high mobility group box or HMG box transcription factors. So at this point, we also looked, Gustavo also looked by ChIP-seq at the ability of endogenous NFAT1, shown here, and the CRIT NFAT1 in either resting or activated cells to bind to across the genome. And this is just an example of what we found. One set of genes looked like this. We only saw binding of NFAT1, the endogenous NFAT1, to the IL-2 promoter, and see that in NFAT1 deficient cells, the endogenous NFAT1 doesn't bind. But oddly enough, cells transduced with the CRIT also don't bind under these conditions to the IL-2 promoter. So this is a family of genes or NFAT ChIP-seq sites that really only likes to bind NFAT with AP1. And in fact, the motif that we pulled out from the hundreds of sites that we got under these conditions was the NFAT AP1 motif, where we have, it's exactly like we saw in the crystal structure. We have a GGA region that NFAT bound, and this TGH, GOC, TCA region that was bound by AP1. In contrast, there was another subset of sites to which NFAT could bind in the absence of AP1. And this is, it can also bind in the presence of AP1, but when we transduce cells with the CRIT, which is incapable of activating AP1 proteins, you can see that they bind in pretty much the same site. And this is the CTLA-4 promoter. You remember that was one of the inhibitory receptors. And we actually saw binding to all of them, to CTLA-4, PD-1, TIM3, and LAG3. And the site that we pulled out from this was this TTT-CCA site, which actually is the same motif, GGAA, the complementary motif. So this is T really, which ought to be up there, GGAA, without a neighboring NFAT site. So we then set up 3 different models to look at T-cell exhaustion. One was the LCMV model that I described before. In this case, the mice are infected with a lymphochoriomeningitis virus, to which they can respond either acutely, or you can make a different virus to which the cells, it's a chronic infection, so the cells become exhausted. And when we looked at, by RNA-seq, at all the different transcription factors that are turned on under these conditions, again, we saw TOX, TOX2, NR4A, and NR4A2 and NR4A1. And that's just shown in a bigger scale here. What we looked at when we looked at for accessible regions, so the only 2 things we did were ATAC-seq for accessible regions and RNA-seq for the gene expression profiles. When we looked at what were high in exhausted cells, the motifs that were enriched in exhausted cells were NFAT by itself and this NFAT AP1 motif, but to a much lower extent. And also NR4A motifs were enriched pretty strongly, and that matched the RNA-seq results. So here are motifs enriched in CRIT NFAT1 induced ATAC-seq peaks. They are identical. You see some NFAT, a little bit of NFAT AP1, and definitely NR4A motifs. So that said that the exhaustion that we were seeing in this LCMV model was similar to the exhaustion that we saw when we transduced the cells with CRIT NFAT. And so the second model that we set up was a B16 over model, where we took a cell that was expressing a B16 tumor. This is a melanoma cell, derived from a melanoma. And these cells have been transduced with OVA so that they are able to present antigen to these OT1 T-cell receptor transgenic T cells. But at the same time, we put in a different transgenic T cell, which recognizes an LCMV antigen, actually. So it doesn't recognize the melanoma tumor. And these were introduced into the mouse with the tumor by these 3 people. Juliana is also from Brazil, Sarah's from Italy. And what we found was that we could find the tumor cells both in the tumor and in the spleen. And only these OT1 TILs, which we could distinguish by this marker on them, showed an exhausted phenotype defined by decreased cytokine production and increased expression of inhibitory receptors. So when we did the RNA-seq, we saw exactly the same things. Among the membrane proteins were these inhibitory receptors, PD1, TIM3, and LAG3. And among the transcription factors and chromatin-associated enzymes that were expressed in OT1 cells, but not in P14 cells, were NR4A2, NR4A3, and TOX. And so basically what we realized was that both for LCMV and for these tumor-infiltrating OT1 cells, we had an overlap with those that appeared in CRIT NFAT1 transduced cells. And more interestingly, when we looked in the ATAC-seq profiles, at what were more accessible in OT1 cells compared to P14 cells, and those are all shown over here, more accessible in OT1 cells compared to P14 cells. When we compared them with an in vitro activation protocol, where we stimulated cells with either anti-CD3 and PMA or PMA plus inomycin, what you can see is all of these accessible regions are shared with the ones in OT1 cells. And if you looked at the motifs that were in these accessible regions, we came up with BZIP family proteins, NFAT family proteins, and of course the NFAT AP1 motif and the EGR motif and the NF-kappa B motif. So these are all activation-related transcription factors that are binding to these accessible regions that are more accessible in OT1, suggesting that the cells go into the tumor, become exhausted, and become activated first. But these 450 regions turned out to be exhaustion-related. They're not represented in activated cells. And sure enough, the motifs that we saw were NR4A family members and NFAT family members. And we're now doing single-cell ATAC-seq to ask if the tumor-infiltrating cells show first an activation profile followed by an exhaustion profile. And the third experimental system we set up was a CAR T-cell system, where we transduced cells with a CAR that recognized human CD19. And this is a CAR that has been used in humans for people with B-cell lymphomas, and has been very successful in controlling those lymphomas. We showed that we could transduce the CAR with 97% transduction efficiency into CD28 CAR T cells, and we generated target cells, including B16 over human CD19, that we put into the tumors. And the volcano plots of all of these, P14 versus OT1, LCMV effective versus exhausted, endogenous TILs versus CAR TILs, show exactly the same pattern, upregulation of NR4A and TOX family members, as well as upregulation of the inhibitory receptors that I mentioned to you before. And this again resembles what we saw with the NFAT RIT mutant. So we then decided, let's knock out NR4A family members and let's knock out TOX family members and see what happens. And basically what we did was take a RAG minus, or in later experiments an immunocompetent recipient, inoculated with the tumor expressing the antigen that the CAR recognized, let them go for 7 days, and in the meantime activate some T cells, transduce them with the CAR, put them into the tumor, and see what happened. And in order to make wild-type cells, we just took these mice that were NR4A flux, 2 flux, 3 wild-type, and put in an empty vector. So there's no CRE. So these are basically wild-type, but they do get the CAR. And to make the triple knockout, what we did was take these minus cells here with the 2 fluxed and put in the CRE such that the flux genes would get deleted and the cells would be triple knockout. And then, of course, we had variants that had 1 or 2 or 3 knockout. And the results were very striking. If we let the tumor grow and didn't give the mice any T cells at all, just gave them a saline injection, the tumor grew at a rapid rate, and these dots that end the cells mean that the mice died or had to be euthanized because they were so sick. If you gave them wild-type CAR T cells, you can see that there is many of the mice die, but some of them survive out to pretty long times, although by 90 days, they're both also dead. If we gave them the NR4A triple knockout cells that had the CAR as well as the CRE, you can see the tumor starts growing, but in most of the mice, there is a regression of the tumor. And we achieved about 70% survival of these mice, which is about as good as you can do with cancer immunotherapy in any tumor, actually better than you can do with cancer immunotherapy in any tumor these days. And the individual knockouts were less effective at this than the triple knockout. And basically, these NR4A triple knockout CAR TILs were like effector cells. They showed decreased expression of inhibitory receptors, increased cytokine production, increased cytolytic activity. And I'll talk about TCF1 if anybody is interested. It's an immunological thing. So basically what this is just showing you that the blue is the NR4A knockouts. And you can see that there are clearly differences between wild type and NR4A knockouts. And all of these differences are good in the sense that inhibitory receptors go down and activation programs go up. So we also looked at TOX. TOX is a member of a very large family of HMG box binding proteins. These include all the SOX proteins that you might be familiar with, some HMG proteins, TCF1 and LEF1, which are important in immune cells. And these 4 proteins in 2007, they hadn't even been named, but it was then recognized that these 3 proteins were similar to TOX, and they've been renamed TOX, TOX2, TOX3, and TOX4. And what these, unfortunately, we don't know what the sequence motif for TOX binding is, because what TOX binding does is bend the DNA without very much sequence specificity. In contrast, TCF1 and LEF1 do bend the DNA, but also show some sequence specificity, and therefore it was able, Rudy Groschedel and Peter Wright were able a long time ago to crystallize it on DNA. So what happens when we try to eliminate TOX? Hyung-Suk Seo from Korea was the person who did these experiments, as well as Adai, who was a student several years ago in Cuernavaca, and came to be, did his senior internship in my lab and also joined my lab as a graduate student at UCSD. So what Hyung-Suk saw was that if he took just the TOX2 knockout mice, they were a little better, but not as much better than than the cells injected with wild-type CAR T cells. So you should be comparing the blue and the green. In contrast, if he injected mice with tox shRNA, and this was partly because this was for two reasons. First, we weren't able to get the tox knockout mice from the people who had it. And second, it was later discovered by people who had used that mouse that complete deletion of tox causes CD8 T cells to disappear from tumors. So tox shRNA is a little bit better than the tox2 knockout, but the double knockout with tox2 knockout and tox shRNA is clearly better than all of them. So in fact, when he did this double thing and compared it to the wild type, where you have tox2 wild type and a non-targeting shRNA, you can see that the tox knockouts cause the tumor to regress with very few exceptions. And once again, we've got 70 to 80% survival. And once again, tox double knockout CAR T cells showed decreased expression of inhibitory receptors, increased cytokine expression, increased cytolytic activity. And this has all been published, so I'll not go into it. And this is just documenting that decreased expression of all the inhibitory receptors, increased cytokine production. So that's been published. So the question was, where do we go from here? And everything I'm going to be telling you about now is effectively unpublished work. So we did accessibility experiments in the TILs that had migrated into the tumors. These are the CD8 T cells that had been given the CAR, given the CRE if necessary, and looked at. And first, I want to tell you that there are a very large number of common things, but when cells go into tumors, some accessible regions go up, some accessible regions go down, as you would expect. And when you look at the accessible regions that are greater in the wild type compared to the NR4A triple knockout, what you can see is that the NR4A motif is lost, just as you would expect, because in the triple knockouts, you don't have it. And the other thing that's lost is the NFAT motif. This is the NFAT without AP1 motif. What is gained in the triple knockout compared to the wild type? I told you that these look like effector cells. And indeed, they show hallmarks of the binding of transcription factors that carry out the activation or effector program. And these are BZIP and NF-kappa B. And we saw exactly the same thing when we compared the tox2 double knockout, which are also effector cells. We saw NF-kappa B motifs come up and BZIP motifs come up. So this is a basic summary of what I've said so far. We know that NFAT with FOS and JUN gives you effective function. We know that NFAT without AP1 turns on NR4A tox. And all three transcription factors together turn on target genes relevant to T-cell exhaustion. There is some crosstalk between NR4A and tox. They sort of control each other. I haven't shown you that. But what I have shown you is that if you look at accessible regions, both NR4A and tox must be suppressing BZIP and NF-kappa B rel transcription factor action, because when you take them away, these are the motifs that get occupied. So that's just summarized over here. And that led us to ask, can we express one or more BZIP or NF-kappa B transcription factors in wild-type T cells and prevent exhaustion? And this was complicated by the fact that there are about 50 BZIP family members, including members of the FOS, JUN, CREB, ATF, BATF, and several other families. And there are at least 5 canonical NF-kappa B family members, P50, P52, REL-A, REL-B, and CREL, as well as some other transcription factors that are actually related structurally to I-kappa Bs, but turn out to be transcriptional regulators. So we decided to do some overexpression in knockout screens, and these were done by Wei Zhang, who is a graduate student in the lab. And he made a screen for these 50 BZIP transcription factors, and he made another screen for the canonical NF-kappa B family members, and the other family members that I mentioned are in process. So the readouts were, which of these transcription factors, when either overexpressed or knocked out, increase NFAT reporter activity in CD8 T cells in vitro? And which of them, a subset of these, were tested to ask whether they increase the numbers of tumor-infiltrating cells in tumors in vivo. So it turned out that from the NFAT reporter activity, I'm not going to show you the details of the screen, BATF, MAFF, and JUN came out as strong hits. And so we tested them in tumors in vivo by taking cells CAR T cells that overexpress these transcription factors and measuring tumor size after the CAR T cells had been transferred. And one of the reasons we chose Jun, besides it's the fact that it came out as a hit in the screen, was that crystal macro, in a completely different system that involved circulating rather than solid tumors, had shown that overexpression of Jun was able to improve the rejection of solid tumor, of those circulating tumors. In contrast, these tumors do not, the solid tumors aren't really capable of being regressed that well by Jun. There's clearly a lower amount than with wild-type CAR TILs transduced with the control vector. But what clearly came out was that BATF overexpressing cells had a very high degree of tumor rejection because the tumor size on day 20 was considerably decreased. In some of these cells, however, some of these mice, the tumors regrew, and so our overall survival rate was about 1/5 of the total number of mice analyzed when the cells were transduced with BATF. And when they were transduced with Jun or MAFF, there's some increase in survival, some delay, but it's very minor really, and the mice all die pretty rapidly. And so basically the summary is that overexpression of BATF in CAR TILs is associated with a pretty remarkable reduction in tumor size 12 days after injection of the CAR T TILs, and also significant improvement in the long-term survival of these mice. So why is this? One reason is simply that the BATF expressing CAR TILs, the CAR T cells, expand in the tumor much better than the CAR T cells injected with the control vector. So a difference of 20% versus almost 80% graphed here. If you, since remember the tumors are regressing when the BATF is CAR TILs are put in, if we normalize these numbers to tumor size, that's even more striking. You have 0.1% per millimeter of tumor, 0.1 into 10 to the 4 CAR TILs per square millimeter. That's, we aren't measuring the volume, we're just measuring one dimension, but many more in the BATF expressing CAR TILs. And that was also shown by mass cytometry using CyTOF. This just shows the whole, we take out the whole tumor and stain the entire tumor with macrophage markers, CD8 markers, and so on. And this gives you the spectrum of cells detected in the tumors, immune cells detected in the tumors by this mass spectrometry method. And you can see the pattern is pretty much the same. Tregs are the same, macrophages are the same, the NK cells are the same, NKT cells are the same, and so on. And there are plenty of endogenous T cells infiltrating the tumor as well, because these are immunocompetent mice. But what you can see over here is that the CAR TILs have expanded enormously compared to the control, exactly mirroring the flow cytometry experiment. So what actually happens? The inhibitory receptors in the CAR TILs are down. So PD-1 is down if you compare this to this. TIM-3 is down if you compare this to this. There's also some TIM-3 non-expressing cells that show up. LAG3 similarly. TOX, which is, I told you, is an important transcription factor in exhaustion, is down. If you look at this double plot, where you're looking at PD-1 and TOX, so PD-1 is the cell surface protein, and TOX is the transcription factor that's involved in exhaustion. In wild-type cells, PD-1 and TOX are going together. In the BATF CAR TILs, the TOX has disappeared, and the PD-1 is only slightly decreased if you compare the MFI. Similarly, ICOS and granzyme B are actually increased in the BATF overexpressing cells, meaning granzyme B is the what CD8 T cells use to lyse tumors, and that's clearly up in these cells, and that's all summarized over here. All the inhibitory receptors are down, TOX is down, Ki67 is up, consistent with the increased expansion. And this was also seen by RNA sequencing. So remember that I told you that 5 of the 24 mice survived, and this experiment, I should say, has been repeated. But this was approximately when the shutdown for the pandemic occurred. And so the mice stayed there and survived for 3 months. And Hyung-Soo came back and re-challenged these mice with the tumor. And what he saw was that aged matched recipient mice would show tumor growth, but each one of these 5 mice showed a decrease. One of the mice showed the tumor, but the tumor regressed, and the other 4 mice never even developed a tumor. And this one mouse that showed tumor regression is actually the mouse that had the lowest number of frequency of CAR TILs in the tumor. So this was very striking because what that meant is that the BATF expressing cells persisted in the tumor for long times, and we've shown that, that's shown here. And because these are in the spleen and lymph node of the mice, because the tumor didn't grow at all, so we couldn't recover anything from the tumors. And when we looked at the phenotype of these cells, they're definitely expressing memory markers. So BATF overexpressing cells can reject the first tumor and prevent a recurrence of the same tumor in the patient. So what was the mechanism that was acting in the TILs? I already told you that there's decrease in expression of inhibitory receptors, increased Ki67, meaning increased proliferation, and increased granzyme expression. But a key mechanism that's acting in these cells is that the T-cell receptor signaling to chromatin is blunted. And this is the TOX gene showing you that. Look, each one of these bars here represents a differentially accessible region between TILs, which I'm not showing you yet, in which PMIG is greater than BATF. And so you can see over here, for instance, that PMIG cells, well, you can't see it yet. If you look at PMIG cells that have gone into the tumor, you can see that between the cells that have been transduced in vitro just prior to their being injected into the mice, and then the cells that have gone to the tumor and then recovered from the mice, what you can see is that there's clearly almost no accessibility in the in vitro cells, but in the tumor, they've become activated through the CAR and probably by other cytokines and so on in the tumor. And you can see there's a big increase in accessibility here, a big increase in accessibility here. If we look at what happens to the BATF CAR TILs, that big increase in accessibility is actually really blunted in BATF overexpressing cells. Other things don't change as much, but these particular things change. And this correlates with the fact that TOX expression is low in the cells. So this is also apparent from this experiment, where we're looking at ATAC-seq instead of, it's the same thing as before, but I'm showing it to you genome-wide. So obviously, when you transduce BATF into CD8 T cells in vitro, you get a lot of accessible regions that are higher in the BATF cells compared to the PMIG cells, and that's shown over here. Each line here is one accessible region. And these are the PMIG cells, and you can see that they don't really, I mean, they share these in common with the BATF. These are overexpressed in the BATF cells, more intense. And these are not there at all in the BATF, unless you transduce with BATF. Now, however, if you look at the TILs, you can see, if you compare the PMIG cells, that the PMIG cells have actually upregulated a lot of things. So there's a lot of stuff happening in the PMIG cells, as I showed you on the previous slide, because they've gone into the tumors. Some things are down, but a very large number of things are up. Now, if you compare the BATF cells that have overexpressed all these things in vitro, and then they've gone into the tumors, they're not really capable of responding. The change between the PMIG cells in the tumor and in the TILs is much greater comparatively than the change in the BATF expressing cells. And these accessible regions, which BATF turned on in vitro, are actually almost disappearing in the TILs. So that says that TCR signaling to chromatin, these accessible regions, is blunted when you overexpress BATF. So one more thing you need to know about BATF is it cooperates with a different transcription factor called IRF4 in many immune cells. And there's no structure of this complex. But what we do know is from Ken Murphy's work is that there are 3 amino acids, histidine, lysine, and glutamate, HKE, that if you mutate them in BATF, they can no longer cooperate with IRF4. And I should also mention that both BATF and IRF4 are induced in activated T cells. So what we decided to do was to ask about the importance of the BATF-IRF4 interaction by retrovirally overexpressing BATF and its HKA mutant in CD8 TILs. The mutant protein and the wild-type BATF protein are expressed at the same levels in vitro. Then they get transduced into mice. You can see that the BATF, this is the same experiment as I showed you before. You can see that the BATF really reduces tumor size, but the HKE is certainly not as effective and it's far more variable. And this small amount of variability allows for a small amount of tumor resistance to the tumor at early times, but then my cells crash. Why do they crash? When we looked at day 20 or thereabouts, you can see that PMEG to BATF expansion is the same as we've seen before, but the HKE cells are gone. They've completely crashed. There are no more CAR TILs left in the tumor. And in fact, if you look at the injection on day 12 and then look at day 16, which is just 4 days after the cells have been injected, they do enter the tumor, but for some reason they fail to survive. And that's also true if you eliminate BATF completely. Either T cells lacking BATF or impaired in this BATF IRF4 binding because of the HKE mutation fail to expand the tumors. In contrast, T cells overexpressing BATF get together with IRF4 and show striking expansion in tumors. And that's already known to be a characteristic of BATF and IRF4 in many immune cell types. So ChIP-seq experiments showed us, so we did ChIP-seq for endogenous IRF4 as well as for endogenous as well as overexpressed BATF because we have the same antibody for both. And when we compared the BATF with IRF4 ChIP-seq peaks versus the IRF4 alone, the vast majority of ChIP-seq peaks that we see show IRF4 and BATF binding in the same place, whether they're the ChIP-seq experiments can only be done in vitro because there aren't enough cells in the TILs for us to do ChIP-seq. We're trying to do cut and run. But at any rate, what you can clearly see here is that wherever there's a BATF peak, there's an IRF4 peak, in the vast majority of them. So what happens when we put in overexpressed BATF? Basically, what happens is that there's a redistribution of IRF4 as well as BATF. And both of them go to these AP1 IRF motifs much more strongly. And IRF4 actually comes away from these ETS-IC motifs. And we can see that by loss of accessibility of ETS family proteins in the tumor. So you can compare the BATF binding from here to here, but you can also compare the IRF4 binding from here to here. There's more IRF4 binding here compared to here. So there's a clear increase. And there's also an increase in BATF at the same sites. Now, you can't compare clearly between these things, but the ratio of BATF to these peaks goes up, as you can see. So basically what we're doing by overexpressing BATF is making the IRF4 work more effectively. So this is almost my last slide. The summary is that T-cell activation turns on two sets of cooperating transcription factors, NF-AT with Fos-and-Jun and IRF4 with BATF. NF-AT with Fos-and-Jun turns on genes like chemokine genes, cytokine genes that are essential, and granzymes that are essential for effective function, whereas IRF4 and BATF are important both for survival and for effective function, as I just showed you. And this is what I told you about T-cell exhaustion before. So we have, in fact, identified B-cell transcription factors, one of them BATF, that helps, that are suppressed by NR4A and tox. I didn't show you those data, and are important for effective function. So by overexpressing BATF in cells that have become exhausted, you can actually improve both survival and effective function in the tumors. And it does this, as I told you, by recruiting IRF4 to composite AICE motifs, thereby promoting survival and effective function. And it also does this by blunting T-cell receptor signaling to exhaustion-related genes and exhaustion-related chromatin regions. And so what are the clinical implications here? The simple take-home message is that targeting transcriptional programs can counter T-cell exhaustion. And a more interesting angle is that we are now collaborating with Yingxiu Peter Wang in the bioengineering department at UCSD, who made a very clever way of turning on the CAR T cells in tumors. What he was worried about was the fact that the CD19 CAR can go away and attack normal B cells in the body besides attacking the B cell lymphoma. And so what he did was devise this method of turning on CAR only in the tumors, so that the CARs can kill tumors only, and he does this by focused ultrasound. So he focuses the ultrasound on the tumor turns on the CAR, and then the CAR, in mouse models, is shown that the CARs can reject the tumor cell, although not completely effectively. But several pulses of focused ultrasound, actually, what they do is increase the temperature of the tumor very slightly by just a couple of degrees. And that turns on a heat shock promoter that drives the CAR. And that's all described here. However, we know that the T cells in the tumor can become exhausted too. I've just spent an entire hour telling you that. And so what we've decided to work with Peter to do is to have the CAR on all the time. Let them go into the tumor, let them get exhausted. But the heat shock promoter that responds to the focused ultrasound is put in front of BATF or other such transcription factors or sgRNAs. They sense, and what you can do is take the tumor that is not being rejected, turn on the BATF, and hopefully overcome the exhaustion program and allow the tumor cells to be rejected as or more effectively. And we could actually do it both ways. We could have both the CAR and the BATF and the T cells under the control of this ultrasound responsive promoter. And so that's all I have to say. My collaborators I have mentioned, I think many times. Gustavo and Renata started the entire program by looking at CRIT, NFAT. This was the B16 OVA model set up by Sarah and Juliana. This was the CAR T cell model set up by James and Joyce. Isaac, who's also a graduate of the Cuernavaca program, helped Joyce in some RNA-seq data. And I forgot to mention that James Scott Brown and Renata actually set up the LCMB experiments about the same time that Sarah and Juliana were setting up the B16 OVA experiments. And the more recent experiments on TOX and TOX2 and BATF were done by Wei Zhang, a graduate student, Hyung Suk, a postdoc, and Edai and Daniela, who are also graduates of the Cuernavaca program. And you may or may not know that they were married while they were in my lab and now have a little baby and are poised to graduate this year. Thank you.
Well, thank you very much. Very interesting talk, very nice. Now the seminar is open for questions. You have questions, you can raise your hand in the participants list, so I can, or just to put your question in the chat, and I can read it, or Anjana can read it for you to answer the question. So far they are not. Well, I have a very simple question. So in all these ATAC-6 experiments in which you analyze the accessibility in the chromatin in the exhausted condition, so you always see that this accessibility increase only in promoters or also
No.
regions around the genome. So what about enhancers? The enhancers that are also overactivated by this particular condition?
In fact, most of the regions are enhancers. What I showed you in the tox locus, for instance, were enhancers that were within the gene body. But there are also many distal enhancers that we can find. The distal enhancers are a little more difficult to interpret because we don't know which gene they're connecting to. But the beauty of it.
Yeah, go ahead.
Yeah, go ahead.
No, you first.
Oh, I was just going to say the beauty of the ATAC-seq is that it can be done on these very small numbers of T cells that we get from the tumors. And they really tell you how cells are responding to the signals.
Mm-hmm. So, and what happens with the conformation of the chromatin in the exhausted cells? Also, there's a clear change in the conformation of the chromatin, for instance, by Hi-C experiments or something like that. Can you detect that?
Yeah, so the problem there is, of course, that Hi-C requires millions of cells, and we don't have millions of cells. But if we look at in vitro transduced T cells that we can get plenty of, yes, any transcription factor that you put into the cell will change the looping pattern around the genes that transcription factor is capable of responding to.
But only around the gene, not in larger places, for instance. The TATs are not.
No, we haven't done those experiments for transcription factors we've used in these experiments. But what we do know is that when other people have done the experiments, there are long-range looping changes as well.
Oh, okay. That's So we have a question from Angelica Santana. Can you please?
Hello, Anjana. Well, a great talk as always. It was very exciting, all your data, changing all these transcription factors and getting these amazing results with the CAR T cells. It was absolutely amazing. I have my question is, what will be, if you have any idea on the signals given by the tumor that makes this turn on the program, on the epigenetic program and transcription program of the T cells? We've been working a little bit on metabolism. Maybe these metabolic things or changes in glutamine pathway or other pathways. Have you got any idea?
I unfortunately have not, we have not done those experiments, partly because the number of TILs are too small. We've thought about doing screens. In other words, let me just turn my video off. In other words, you know, it's clear that they go into the tumor and turn a bunch of things on, right? Accessible regions change, RNA expression, I mean, gene expression changes. So the question is, what inside? Is it metabolites in the tumor? Is it a different metabolic environment? Is it cell surface contacts between cell surface receptors and their ligands? Is it simply antigen recognition? You know, what exactly is going on? And the answer is, unfortunately, that we don't know. Okay. And the only way we would actually be able to find out is either changing things one by one within the tumors, which seems to me to be a little difficult.
Yes, absolutely. Yes. Well,
doing something high throughput.
Yes.
Where we can, where we can send TILs into tumors, knock out every gene possible at, you know, all the genome-wide screen. But then the difficulty would be getting the T cells back and saying which gene got which shRNA and how did it change. And so we haven't actually done that.
Okay. And the other question is, there were some very old publication on the types of NFAT, NFAT1 versus NFAT2 into the way they behaved in the very early days about Th1 towards Th2 that you published actually. And I wonder if you know if there is a difference between the NFAT that binds to Fos-Jun and the NFAT that induced the exhaustion program.
That's a very, very good question. I'm not working on this. My collaborator, Patrick Hogan, is. But I know the data, so I can tell you briefly that the DNA binding domains of all these things are the same. So all of them would collaborate with AP1. But what he's been able to do is to knock in this RIT mutation into NFAT2. And what he finds is that NFAT2 does not really affect the exhaustion program. It's more involved in the activation program. So the reason people have attributed this big role in exhaustion to NFAT2 is simply because it's induced in these cells before they become exhausted. But when you actually look at it one by one, between NFAT1 and NFAT2, there's no change. I mean, the NFAT2 is not doing anything. NFAT1 is the one that's doing something for the exhaustion program.
Okay, but because we found differences between neonatal cells, neonatal CD8 T cells and adult cells, and mostly neonatal cells were in the active enhancers of neonatal CD8 T cells, we find mostly NFAT2, whereas in the adults was NFAT1. And I thought it might be interesting, these differences in usage of, but anyway.
Between those two are really, really interesting and require more analysis.
Yeah, well, thank you very much. Great talk, as always.
We have a question in the chat. So from Dr. Luis Covarrubias, you want to read it or I read it for you?
Yeah, I'm happy to read it. NR4As are considered orphan receptors. Is there a possibility of endogenous ligands or the possibility of developing artificial ligands that can control NR4A transcriptional activity? So in fact, there are papers that show, basically have generated by screening artificial ligands against NR4A and have shown binding of those ligands in structural experiments. It's not clear to me that anybody's actually used them clinically, though. But during the pandemic, there was going to be a conference on NR4A proteins, just NR4A proteins, nothing else. And I was going to go to that conference and see if I could find these people and talk to them. And but because of the pandemic, I didn't do that. So it's a really good question and a really good time to revisit it.
Okay. More questions? So I have another one. I don't know, it's quite general. And following your talk yesterday with the students, in these exhausted cells, what happened with the TAT proteins and with the 5-hydroxymethylation of cytosines in the exhausted cells?
So we've done an interesting experiment, which doesn't exactly answer your question, but basically we've taken the TET proteins, and these are TET fluxed cells, and have introduced the CAR into them. And they definitely reject the tumor better. And in fact, there's a paper from Carl June that says that there was one patient into which they introduced a CAR, and the CAR just by accident integrated into the TET locus, and its function was lost. And the other allele of the patient was a hypomorph of TET. It had a mutation that they didn't know about. This patient rejected the tumor completely and is now alive and doing very well 5 to 7 years later.
Oh.
So that was the reason we tried the experiment, and they definitely, and what we think is happening is that, remember I told you yesterday that loss of the TET proteins gives you inflammation. And what we think is happening, the TET proteins are inducing an inflammatory response in the tumors and maybe recruiting and improving the immune response to the tumor. So
very interesting. So we don't have further questions. Let me check the chat. No. Well, Anjana, thank you very much. It was an excellent talk. We learned a lot, and I'm sure also the students in the program. And I'm sure that we are going to keep in touch with you for the future, and probably many of the students too, very soon.
Yes. Thank you very much.
Thanks for the time.
Thanks.
Okay. Thank you.
Bye-bye.
Bye-bye.